exciting session. You're welcome to come in and have a seat. We're going to be just a few minutes to start, but I wanted to talk to you about something that's a DLD spirit and a DLD tradition, and very much inspired by Yossi Vardi, which is one of the amazing founders of this event. This event, as you know, is about all the people around here. It's hand-curated. Everyone here are entrepreneurs, innovators, investors. Many people around you might be your new partner or your new investor or your exciting new investment. So what I would like for everyone to do, it's a small chore, but it's very rewarding, is to look around you. Look for someone that you have never met before that happens to sit next to you. Get to know them. You have 1.5 minutes to do that, and it will be rewarding, so go. Introductions are taking place. We're ready to start. So we need everyone's focus on the stage. A lot of interesting things are going to take place right here. You three especially, we're starting. Thank you. So as a person that's coming from a software background with I'm Tal, by the way, if I didn't introduce myself. I'm Tal, co-founder and CEO of Evolero, hyper-social event websites, powering this event as well. I'm into software, so makers is something that's very exciting for me, and I can't wait to see what's going to happen here. So without further ado, I'm going to invite our speakers for today. First one who's going to moderate the session is Professor Ronit, Satchi-Fainaro. Welcome. She's from, you can receive her. Ronit is the head of vascular biology and theranostic nanomedicine research, laboratory president of the Israeli Society of Controlled Release. Welcome. I'm going to invite the other panelists, but they're going to be giving you their presentations one by the other, so they're going to go up to the stage and we'll start gradually. So first one I'm going to invite is Yair Moshe. Yair is a senior engineer in the Signal and Image Processing Laboratory in the Technion. He's currently pursuing the PhD in computer science at the University of Haifa. Yair has been actively involved with more than a decade in research and education in the areas of signal processing, image processing, and computer vision. Next one up is Guy Hoffman from the IDC. So can robots and humans interact the way that human beings interact? Sorry, can robots and humans change the way that human beings interact with each other? Guy Hoffman researches embodied cognition and intelligence in robots. Next one up is Yonatan Wexler. Yonatan is missing, so he might join us very soon, and I'll introduce him when he's here. We're going to start, and the first presentation. Sorry, you'll go ahead and start, and then I'll call the presenters one by one. So you can go right up to the stage. I'll tell you a bit about Yonatan. He's an experienced computer vision scientist who has worked at Oxford University, the Weizmann Institute of Science, and Microsoft. He has spent the last 3 years developing a unique device for blind and visually impaired people. One of the novelties of the device is a unique user interface which interprets the user's gestures. By simply pointing at what he or she wishes to know more about, the user engages the device, called OrCam. you'll probably hear more about it soon.
Okay, thank you very much for the kind invitation. This is not really my milieu. We were talking about funding. I'm very good at spending, and you'll see very soon how do I spend. And my main spend is on nano-GPS for cancer theranostics. Theranostics in general is a very hot field in medicine in general and specifically in cancer. This is the combination of therapeutics and diagnostics together. So my work at Tel Aviv University at the Department of Physiology and Pharmacology is based on a fascinating paper that came out in '93 on 16,000 autopsies. This is something we can only dream about in Israel in terms of a clinical trial. And these were autopsies of people that died of trauma. They died of a car accident, suicide, anything but any diseases. And you can see the striking numbers here. What they found was that 39% of the women were found to have these tiny lesions, tiny cancers in their breast. 46% of the men had these tiny, again, lesions of cancer in their prostate, and virtually all, 99.9%, had these tiny lesions. And when I say tiny, they are about 1 to 3 millimeters in size, these tiny cancers in their thyroid. So now everybody is depressed and I killed all the atmosphere. But the more optimistic view is that only 1% of the population, those populations that are clinically diagnosed, are diagnosed with breast or prostate cancer, and only 0.1% are diagnosed with thyroid cancer. So that made us very curious to think, why is this discrepancy between the really high numbers, meaning that all of us have these tiny lesions in our bodies, compared to the ones that are clinically diagnosed? And for that, we call them, those tiny lesions, as dormant tumors. It's not that they are asleep, they are very active. They sit there, they proliferate, meaning that they divide, but at the same time they die. So they keep a certain amount of balance between the cells in our body that die and proliferate, and they remain at this size of 1 millimeter without being able to recruit blood vessels that will feed them. The blood brings with it all the nutrients, all the food, all the oxygen that every tissue, every organ in our body needs in order to grow in size and progress. So what we can see here is that at some point there is a tilt in the balance between those factors that recruit blood vessels to those that prevent the blood vessels from growing, and we don't exactly know always what is this trigger, but once there is this imbalance, they start being able to recruit blood vessels. Once they recruit the blood vessels, they get all the food they need and they grow in size. And not only that they grow in size as the primary tumor, but they can now send the tumor cells back into the circulation, into the blood vessels, the new blood vessels that were formed, and they will circulate in all our circulation and get out of the blood vessel into a distant site. And this is what my lab mainly works on, is this metastasis. Because most people don't die from the primary cancer. For that we have wonderful surgeons that can just resect the primary tumor. The problem is when we have a systemic disease, meaning in many locations, we have the metastasis spread around, and the way for them to spread around is using those blood vessels. So these dormant tumors that were found in these 16,000 autopsies basically were manifested as primary tumors that none of us know about them because they are asymptomatic. We don't feel them, they don't interfere with our daily lives. We can live with them until the age of 120 without even knowing that they are there if we don't have that event that triggers them to switch to recruit blood vessels. The other stage is once they already circulated in the body and went to a distant site, when they remain, again, dormant for a very long time. And the question is if we are able to image them, because this tiny size of 1 to 3 millimeters is below the detection limit of most current imaging modalities, and we will talk a lot about imaging today and image analysis. So what it looks like is once we have this mass, tumor mass in the beginning, then it starts secreting those factors that are called chemoattractants, meaning that can attract chemically those blood vessels that are pre-existing in our body, are normal. They never divide. They don't have any reason to divide and grow. It's the blood vessels that we were born with. These are the blood vessels we die with. The only thing, they are quiescent. They are sort of dormant. There are only 3 cases when we become adults that they start growing when it's not pathological, is in the time of wound healing, or when there is the menstrual cycle, or in pregnancy. But other than that, there is no reason for blood vessels to be created. So why is this interesting for us? We have about 19 billion blood vessels in our body, capillaries. They can, basically from each one of us, they can circle the earth twice, or if we take a tennis court, they will fill in the whole tennis court just from one person. So probably they have some importance to their function. And we would like to know what are the basic mechanisms that maintain the state of tumor dormancy, meaning keeping those 2 tumors dormant. Because if I could read that original paper saying that, oh my God, all of us have cancer, but on the other hand, I can think that we have something endogenously, inherently in our body that keeps them dormant until that trigger. So if I can mimic what are those factors that keep them dormant without being able to recruit the blood vessels and find them early enough, then we can keep them dormant for all our lives and it will not bother us. So this is why we start from basic science. Then we look at the events that initiate the transition from dormancy to the progressive disease. We look, is that process reversible? And we would like to define the mechanisms that cancer cells recruit normal cells from the microenvironment, recruit them to their own benefit. They are sort of parasites because they alone will not be able to grow beyond that size of 1 millimeter. They start growing only when they recruit the blood vessel cells, those are called endothelial cells, and other normal cells that they hijack their functions to their own benefit. Once we will figure out the first 4 questions, which probably will take all my career, then we can apply this knowledge sensitively and to detect and treat cancer by disrupting these interactions between cancer cells and the environment. In every tissue, and every tissue cancer tissue that I get from the surgery room, I see that there are only about 30% of cancer cells, about 70%, 50 to 70% usually, are cells that are normal and they were just recruited by the cancer cells to hijack them. So how do we address these questions? We start, like many of us here, with models. We create models, 3D models in the tissue culture, in the lab. Then we create them in mice, in animal models. We compare. Biology is medicine. This is why it's called the art of medicine or medical practice. It's not exact science. We have a lot of averages. So we would like usually to compare. We compare cancer tissues to normal tissues. We compare very aggressive tissues, cancers that were able to recruit blood vessels to the ones that are dormant. And we compare their gene signature. We look what is different, which genes went up, which genes went down, RNA, and so on. And we look how it affects their function. Then we'd like to reverse everything and take it actually from the clinic. Start from the person, from the clinical trials, from the surgery room, take it back to the lab, and take from every patient, this is personalized medicine, to see which drug will fit him best. And for that, what we are doing is we're taking the clinical specimen from the surgery room. We do the image analysis from the MRI, and then once we have that, we can put it in the software for 3D printing. So most people in biology or medical engineering would like to do artificial organs. Before, as I tell my kids, before you ask me to buy a new toy, try to fix it. So before I try to replace the organ, I would like to fix it. If we have a tumor in the brain, I would like to make sure that I can shrink it back to dormancy and in a way fix the brain. And for that, my 3D printing is actually printing cancer. I'm trying to print cancer from a patient and then immediately give an answer with our nanomedicines. Can it affect that cancer or not in a real time, that it can actually affect that patient and not only 20 years later, the next generation. So we have several polymers in the lab that we synthesize. These are nanometric. They are around 20 to 30 nanometers in size. And the importance about that nano size, we always ask what is this nano shmano that everybody is talking about. So nano in general is 10 to the minus 9 meter. And when we talk about nanomedicine, we talk about usually 20 to 100 nanometers. Why is it important? If I take the analogy to a street with a normal street and then there are turns, if I'm a person walking in that road, I can turn when I have these turns, these leaks in the way, or I can just go up on the curve. But when I take this taxi, this Waze taxi here, and I put all my passengers inside, and my passengers are DNA, RNA, different drugs that will attack the cancer cells or their environment that supports them, they can only get out from the road in those turns. They cannot go up on the curve. That means that nano, for us usually, it's very small, but for our blood vessels, this is huge. And normal vessels, as you can see here in the upper panel, normally they are very thin, very regular, and what you see here, I injected our polymers that are shining light. When you go 2 millimeters aside, the bottom, this is osteosarcoma, a bone cancer, you can see that everything leaks outside. This is just physics or mechanics. The nanoscale can leak out only from those new blood vessels that were created very quickly and are leaky, but they cannot leak from the normal vessels. Why is it important? Because the main problem of the current chemotherapy, classical conventional chemotherapy, is the side effects. And as I said, the problem is the metastasis. It's a systemic disease. We need to give chemotherapy intravenously so it will get everywhere, but it will get everywhere and cause also the side effects. And in this case, it will leak out only from those abnormal vessels in the tumor. And these are how our multivalent polymers look, and we can hang on them whatever we want. As I said, different drugs and different fluorescent probe at the near infrared and wavelength. And I'll show you in my last slide how it works. For example, we have a trigger, which is linked to our large or tiny, how you look at it, polymer. And there is a trigger, and that trigger is cleaved by one of the analytes or one of the proteins that we saw in that original signature, that it is overexpressed in the tumor. So we will inject it intravenously, and within 2 minutes, it will, from being turned off, it will turn on, shine light only in the tumor where it sees that protein, shine the light and tell us that not only it detected the tumor, but also released the drugs at real time. So the impact of this research is that we will get a new definition of a universal dormancy signature, what means how the gene signature looks like. We can, in the translational level, we will be able to target new nanomedicines that will be non-toxic because it will not go to the normal healthy tissues and will not cause side effects. And in the clinical aspect, we will revolutionize the way we think about cancer. If we look back, this is Fleming who invented the penicillin 100 years ago. All we could have done with an infection is amputate the organ 10 centimeters above the infection. Now we can take the antibiotics without even knowing where the infection is, and it will treat it and become very quickly, within 2 days, asymptomatic. This is where we want to see cancer in the next 10 years and basically transform cancer into a chronic yet manageable disease. This can only be done with a multidisciplinary lab coming from biology, medicine, engineering, and chemistry. And it took a while until they talked to each other and understood, because these are different languages. But I think if we drain the water from the ocean, we will all find out that all the islands are connected. So thank you very much.
So next presentation we're going to see is from Yonatan Wexler. come right up.